So the people can hear me?
Yes.
So the recording can hear me?
Yes.
All right. Can we start a recording?
Yes.
No. Have we? All righty. Oh, look at you. I'm so impressed with you. All right. So, thank God for post-processing. All right. Hello. Welcome to the next generation sequencing seminar series posted by the IUPUI Bioinformatics Club. This is our first session in the series. So this is just gonna be like an introduction to NGS and some of the other tools and things associated with it. It's fine. Maybe. All right, there we go. All right. That was a bit of an introduction. So information for life. This has to move. We're gonna be constantly moving this, and hopefully like over here will just be okay-ish. All right. So information for life is stored in large macromolecules composed of like smaller units. So in general biology, there are three classes of molecules. Macromolecules specifically, so DNA or deoxyribonucleic acid, RNA, which is ribonucleic acid, and then proteins, which are polypeptides or strings of amino acids. And they vary wildly in length from being say pretty short to only being like tens, or just above 10 units, to being tens of millions of units, like for chromosomes. And then because this will be relevant later, I'm just going to kind of show how the bases. So here's a basic chemical reaction for how you have a DNA base. Like so you have DNA and it has a base at the end, and then there is a triphosphorylated group attached to the next base, and then you just do a hydrolysis reaction. And you to form like this diphosphate, and then you just have this glycophosphate bond between those. That kind of clear? Okay, this will be important when we later. All right, and we're okay. Move this down a little. All right. So next generation sequencing, or NGS, is just like the most recent technique for like biological sequencing. So if we, this is a nice time graph going from like 1980 to relatively present day. And if we go back much further than this, it just drops to zero. And so this has largely been broken up into like three distinct phases. So the first set coming out in 1980-ish is this gel-based systems. So they used polyacrylate or agarose gels, and you could kind of crudely separate molecules by size. We're not going to go too far into it, but so you take the molecules and you inject them onto the gel, and then you run a current through the gel. And then because these molecules are polar, they'll move from one node to the other node. And then the rate at which they move through the gel will be based upon their size. So you could, like I said, rather crudely separate them by size, and then you could excise them from part of the gel, and you could do some like chemical sequencing and try to do some chemical tests and try to determine what the composition of that was. And that was like the very beginning. And then gradually, like you got better at doing things like that. You could develop different types of gels that allowed for more better separation, and you could gradually just get better at identifying what the pieces were and separating just individual pieces. Then we have a second stage, which is this capillary sequencing, sometimes also called Xander sequencing. And you could use modified nucleic acids and to determine like the sequence of the molecule that was contained in the in a very small capillary. And then by doing a bunch of these sequencing experiments, like this small fragment and this small fragment and this small fragment, you could eventually get enough fragments that you could start to line up the fragments and put them together. So that was this capillary sequencing. And then finally, we move on to like this massively parallel sequencing. And this is, so we're still using modified nucleic acids, but this time we're also using some modern analytical platforms and computational tools. And I'll go over all of that a little bit more. But the other important part of this graph, as you can see, is that the number of kilobases that you could analyze per day has just exploded exponentially. So, in the early days, you kind of had a linear growth from over 10 years. It went up from 10 kilobases today to like 100 kilobases per day. And then with capillaries, it got a lot better. We went up from like 1,000 to a little over 10,000, which again is great, except that we're talking about chromosomes that are millions of nucleotides long. And you just say, imagine putting together imagine I have a phone book and I shred the phone book into a bunch of little pieces and then say, all right, here are the pieces, put the phone book back together. Like that's really the scale on which we're talking about, until we finally hit like this massively parallel things. And now, you could have, so if you guys are familiar with like the Ancestry, so you can largely, like for $1,000, you could have your whole genome sequence. Ancestry cheats a little bit, and so they only check a couple of key gene markers, and they just use those, and that's why Ancestry is so much cheaper than like actually having your full genome sequenced. All right. For the archives. All right, so in the modern space, we have 3 platforms, soon to be 2 platforms, and I'll explain that briefly, that have really arisen as like being the best in the business. So Illumina, PacBio, and Oxford Nanopore. Here we have, so you can see, ignore Ion Torrent. It's here for completeness, but no one uses it. It's just you can look through the numbers and kind of recognize it's not nearly as good as the other 3. Also, these are old figures from 2014, but you can see so the way that Illumina does a lot of reads, so 300 million reads per run, but their reads are relatively short, so they can get 2 150 base pair fragments and then just copy those a bunch of times. Whereas PacBio and Oxford Nanopore are both trying to do, so like the idea is a single read. So here's your RNA or your DNA molecule, and the idea of PacBio is we have a modified protein with a modified nucleic acids, and so we have it will duplicate the thing that you give it, and then as it's duplicating it, it will grab one of the modified amino acids and cleave off one of the, this fluorescent tag. So then it will briefly like fluoresce and let you know like and each base has a different color. So then by going back through what the flashes of color were, you can then determine the sequence. Oxford Nanopore is similar in design. So it takes the single molecule and runs it through a modified ion channel. And so as the bases go through the ion channel, it changes the voltage of the ion channel. And so by analyzing the voltage and then use some machine learning, you can say what the sequence was. As you can see, with a bit of let's say a bit higher error rate than the others. Illumina is a bit more complicated, and I'm not actually going to go over it too much. But the idea is, we have a probe here, we have a probe here, and we take our DNA, and it will latch. It starts in two locations, and they'll latch onto each other, and then we'll cleave them, and then we'll run nucleotides down there. So that the first nucleotide will bind to this location, and then we'll wash it. And we'll wash all the other ones, and then we'll check, and then we'll add the next nucleotide, and and just repeat that process iteratively. And so that's why it requires, like, it uses short reads, because that's just like the bridge gap of these things. And then it sequences them a whole bunch of times, because you have a whole bunch of copies of like a relatively short fragments. So that is the this is the picture into the modern equipment and the general gist of how those theories work. You can go to their websites and you can learn more about how those work. Also, Illumina bought PacBio a couple of years ago, and so it's unclear what they're going to do with it. If they're going to continue to make more PacBio systems, or if they're going to try to marry the two, or come up with some other new technology. It's all up in the air right now. All right. And now I'm just gonna go over so we're gonna start with some of the basic file formats that are used for these platforms. So the FASTQ file is the basic Illumina file. So when they take the data that they got from their experiment, and they read them all into reads, and those reads are composed into this FASTQ file. For the PacBio results, it's called a BAM file. This will when we go later, not today, but in the next section, we'll talk more about BAM files. Yes, they're called the same, they're the same file format. They're called the same, they use the same architecture, but this is typically called an unsorted BAM file. So it hasn't been aligned, it hasn't gone through the steps that usually are associated with the BAM file. Yes, I reckon it's a little confusing. You just have to be careful with your naming convention. And then finally, the FAST5. And this is the traditional nanopore format. And I'm not going to say it has all the sequence information, but it also has some other information that exclusive to nanopore and how that technology works. So we're not going to go over it. Just know that if you see a FAST5 file, it's from nanopore. It's from nanopore sequencing. So then the next question is, well, how do we get this data? Where do we go? And it's well, so there are three, depending on where you are or where you want to get the data from, there are three big sources. So in the US, we use NCBI, which is the National Center of Biological Information. Specifically, you can use the GEO, which is the Gene Expression Omnibus, or SRA. Yes. So if you're in Europe, you want to use the EMBL, which is the European Molecular Biology Library. And then there is the DDBJ, which is the DNA Databank of Japan. And as a note, NCBI, EMBL, and DDBJ all share information as part of just a global consortium. So any data that is given to NCBI is then shared with the other two, and vice versa. So you have the 3 different portals for regions, so that you don't have. So this was started in like the early 2000s. So or actually even before that, in like in the late 90s. So like high speed wasn't really a thing, and so like pinging one server in Europe would take, you know, minutes every time you wanted to do something. So it just made sense to separate them out geologically. And it's just, at this point, there's infrastructure built for all 3 of them, and no one wants to, like, you know, give them up. So yeah. All right. So this is like the traditional pipeline for NGS analysis. So there are a variety of different steps, and then at each step there are a host of different tools. Like I've listed just the ones that like I'm familiar with and that I've used personally, but this is by no means exhaustive. So if you say, you know, you can do a quick e-search of say of any of these words or any of those tools, and it'll come up with a list of other tools. So typically the first thing you want to do is you want to analyze once you've gotten your read data, the first thing you want to do is analyze it for quality. The traditional tool for this is FastQC, and that's what we'll be going over later in the demo. Then once you've done the FastQC, you can then trim the adapter sequences. You can use using FastX, Trim-O-Matic, or Trim Galore. And we'll talk. So the adapters are as part of like you'll recall, like for Illumina, I talked about their like the 2, like they bridge. Well, they bridge them using the using adapters. And so you need to remove those adapters to just get the sequence that you're looking for. Yes, so there's a different pipeline for those. Most of the data that people are using is Illumina. It's just the cheapest and easy to say and like easiest to analyze. So that's what we're gonna focus on. But good point, Sawyer. So then after that, we want to align if we already have a reference genome for the species of interest, which is typically the case, then we want to align our reads to the genome. We can use HiStat2, STAR, or if you're feeling old school, you can still use Bowtie. But suppose that you were ever in an instance where you didn't have a reference genome and you were assembling it yourself, then there are a couple of assemblers that you can use. Canoe, Velvet, Plas, or Spades are all all tools that are either relatively new or are still receiving updates. And then finally, depending upon what your protocol is, you can do things like peak calling. There's Piranha and Max2. And then the final thing I want to talk about. Sure. So I was gonna, can I go through this and then yeah. All right. So the last thing I want to talk about is like the different types of NGS experiments will generate different data. So depending upon what protocol they used in their study, we'll give you different information, and then that will determine like what you really want to do with it. So most basic is DNA-seq, which is just DNA sequencing, and like you're just trying to, like when you do this, you're just trying to determine what the genome sequence is. So this would be like one of those early when I said like de novo assembler, like you'd be using DNA-seq data. RNA-seq data is RNA sequencing. So this is a way of determining which genes are being expressed and at what levels are those genes being expressed. We have what's called chip sequencing. Yes, that looks weird, but that is the correct punctuation for it. So this is a type of DNA sequencing, and specifically, it's a type of DNA sequencing where you can determine where a particular type of protein or proteins are bind to DNA. So this is what so when I'm talking about peak calling, this is like something like ChIP-seq or ATAC-seq or RIP-seq or POP-seq are the things that like you're that we're using it for. So when you end up with a bunch of reads that will kind of like they'll all be like some of them will be different portions of the DNA that it's bound, like just different signals, and then you'll get different ones at different frequencies. And so you can do peak calling to like try to fit like a Gaussian distribution on those, so that you can kind of determine where, let's say, where is the true location for where this protein, like the most likely location this protein is binding. So, you know, is it recognizing this motif, or is it recognizing this motif, or is it recognizing this motif? Like, so you can use peak calling to try to determine that. So the next section is ATAC-seq. This is a special type of DNA sequencing where you can determine open regions of chromatin. So basic, I guess, a basic molecular biology. So we all know DNA is like this double helix, and we kind of just like think of it as like it's just like this double helix in the cell. But if that were true, like we would be very, very like cells would just be very, very, very long. Like if you, I don't remember the statistic, but it's like if you lined up all of the DNA from what's contained in a cell, just like from bottom to top, it like goes over the Eiffel Tower. So it's not, that's what like it can't be stored practically that way in the cell. And so instead, it will spiral itself into very tight, very compact configurations, and will only unwind parts of it to make them accessible for proteins or for other things to recognize. And so ATAC-seq is a way of trying to recognize what sections of the DNA are open and available, as opposed to which sections are like tightly coiled amongst themselves in other complexes. So RIP-seq is like the cousin to ChIP-seq. So instead of seeing where proteins are bound to DNA, you can see where a particular type of protein is bound to RNA. And then a relatively new technique that was created here at IUPUI is POP-seq, which is a type of RNA sequencing that tries to determine, like in a global, like transcriptomic level, where are all the places that proteins bind to RNA. So like where RIP-seq is like one particular protein or type or like family of proteins with like very similar structure, POP-seq is like everywhere. It's like all of them. All right, so that's all for our introduction section. So we'll be moving on to the demo. And so I'm just going to help lay out what we want to accomplish with the demo. So we're going to be going over like the registering for the Carbonate computing accounts here at IUPUI, if people haven't done that before. We'll also be downloading PuTTY and WinSCP for Windows. If you're not using Windows, you can just use like traditional command lines, SSH, things like that. Where you can get how to download the data from SRA. We'll use PuTTY to log into Carbonate. We'll load a FastQC module on Carbonate, and we'll just run FastQC. And then we'll transfer files to and from Carbonate with WinSCP. And then we'll finally get a chance to look at our FastQC results and talk about the various things that they're analyzing for. All right, I'm going to hand off to Neil, and you can start the demo.
Okay. Thank you, Xander, for giving us the background on NGS.
To get started, I would ask everybody to open up your computer and let's get the Carbonate account registered if you haven't already. And I will follow along with you guys and show you how to get to that. Account. Okay. Yes, you'll have to log into the account. If you don't already have it, the Carbonate, then follow me to get registered first before we start downloading PuTTY and WinSCP. So go to this Get Additional IU Accounts website. Oh, it's not shared. Xander. One, sure, yeah. Still not shared? Okay, let me share. Okay, should be shared now. Oh, it's not duplicated, okay. Okay. All right, so go to Get Additional IU Accounts on Google and then log into your IU account. Once we're here, scroll down to go to the Create More Accounts. And that will lead you to the accounts here that you can access. So there are different types of there's Carbonate, Big Grade 3, Karst, Slate Project, different groups that you can have access to. So everybody has access to this? Okay. How far is everybody here? We need Carbonate. Already have programming class. So do you already have it? Yeah. Okay, perfect. And Anna, you have it, I suppose? Okay, perfect. Yes, so society, let us know once you're registered. It may take a minute to like give you an access. It's automated, I think. But in the meanwhile, everybody has access to PuTTY, and so now what is next? Yeah, PuTTY and WinSCP. So we have PuTTY installed for Mac. We don't have to. You guys don't have to worry about it. WinSCP, Mac people can use WinSCP for better. Yeah, yeah. So okay, for Windows, I think Anna, you're the only one who's with Windows. Yeah, wow. We have a lot of Mac people. Xander, we may need your assistance here in Mac. Okay, and then SRA. So Xander, I'll bring you in here for the SRA again, for the data set. But I want to make sure that everybody has it. So I am going to log into the PuTTY real quick. So when you open PuTTY or your terminal, you want to go to your Carbonate one. So it's carbonate.uits.iu.edu. Yeah, yeah. So you'll be good for the next session, where we'll do a lot of hands-on. So, okay. So log into your terminal. Okay. Oh, there was a there was a game. So they were teaching us how to use the supercomputers and all the different ways to access it. So they made an interactive game. So you learn all how to do how to submit scripts and everything. So it was fun. Okay, let me get this one. Should be good.
Yeah. So you. This is what the terminal looks like, and again, I want to show you guys the PuTTY as WinSCP as well. Okay.
Yeah. You
Just for visualization, but so it's carbonate.uits.iu.edu. So this is your home page when you open the Carbonate. I already have a couple folders, but so let's look at PuTTY. So once you log into your terminal, you have to first make sure for Max people, it's again carbonates.uits.iu.edu, and you have to log into your paraphrase, which is your IU account. Once you give your credentials, it will pull in this terminal, and it will automatically load in some of these primary. So I don't know, is it the same for the Mac? It automatically just loads some of these. Okay, so first thing you want to do is how to clear the screen is to Control L, which is just clears the screen. So now we can start fresh. So we want to learn all the modules that's available on the Carbonate. So the system itself, the administrators who is maintaining the Carbonate, they have installed multiple tools within Carbonate. So how to access that? So module avail, so that means module available, that will pull up all the different tools that is installed in the Carbonate system. So what we're interested in is the life science one. So scroll up and look for this Carbonate life science section. And here we will be using a lot of these tools are applicable to the NGS pipeline. So for example, we talked about Canoe. So these are Canoe, which is for the de novo assembly. Blast, bow tie, high. Yes. So we'll be using FastQC today. There are different ones default as well. Yes. Matic, trim galore. Yeah, these two. So these are all NGS pipelines that we'll be using tools in the NGS pipeline. Cut adapt is another trimming tool. Bad tools, same tools we'll use. Yeah. Okay. All right. So just giving you the introduction of all the different tools available to us. So now, going back to the presentation. Now we want the data, right? That we're going to do preprocess or FastQC for the quality check. Right. So before that, let's do that. Let's see. So we're going to get our data from BI. That here. From here, we're going to SRA. Okay.
Sorry. There are a couple of ways of getting information. Every experiment has its own SRA ID. So if you happen to know that, you can just type it in here and pull up that information. Or if you don't really know what you're looking for, but you don't know what the ID is. So it's like, we're looking for zebrafish and we're looking for RNA-seq. So you can see it'll pull up all of the. So it pulls up 46,225 results for those things. You can also come over here and filter by taxon. So we can see here, of those 46,000 results, 45,364 of them are from Danio Rario, which is the Latin name for zebrafish. So then, but we can select that. And now, as I say, so now it's added this this tag up top, but it's now all of these are just from that species. So all of these are zebrafish ones. So we could peruse around, and as we scroll down here, you can see. So this is click ITSec, which I don't know what that means. So learn something new every day. But this is from, so it's an Illumina. It's from the HiSec 2500 series. So 3.5 million spots, 263.8 million bases, and then 96.2 megabyte download. So this tells you how many reads that they got, how many bases were contained in those reads, and then this is how large the file is if you try to download it. And say, as you can say, here's the accession number. So like I said, each one has its own accession number. The one that we're looking for, or the one we're going to use for this series, and the reason that we're going to use zebrafish and not humans is that zebrafish are, say it's a standard model. It's called a model organism. There's a whole bunch of data that's been done on it. People study it all of the time for like a host of different reasons. And their genome is just way smaller than ours. And that means that we can reasonably we should be able to reasonably get all of the steps of this done without having to actually queue jobs on the supercomputer and wait for those to get done. And we really want this to be a more interactive series. So that being said, oh, that's all right. So SRX 1382 3180. Okay. So this is so when we click on one of the experiments, it'll pull up here and it'll just give us more information. So so this is specifically mRNA. It was submitted by this by Yale Geraldez. Here's the study, and so typically it'll have so here's the abstract for how the data say, and that tends to say what was the experimental protocol for this experiment, also why was this experiment done. If there's a paper, there'll also be a link to the paper, so you can go there. And then it'll also tell you, so here's the more information about the library that was used for this analysis. Yeah, that's okay. So then we're just going to click over here on the run tab, and it'll pull up so this is the meta information for this file. And part we're interested here is so this is the metadata. You can come over here to analysis, and you can come over here to the reads. But the one we're interested here is the data access. And if you want, I'm not going to go, we're not going to go over this right now. So this specifically, this is paired data. If you want the whole data, we could just put in this, this hyperlink, but we're just going to only use one of the pairs for this. So I'm just going to copy this, and I'm going to go to my I'm going to go back to my terminal on Carbonate, and I'm just going to hit wget, and then I'm going to insert, I'm just going to paste that address that I copied, and then I'll just press return, and it will download this. So I would just go to, let's say I would go to Slate. So /n/slate/ your username.
Carbonate gives you 100 GB of storage space. Slate gives you about 10 TB of storage space. So depends on where you want to save it. So we just logged into Carbonate. We can still access Slate from this.
Okay.
Yeah.
So you have to have an account with Slate, though, because I don't have one with Slate, I don't think, so.
Yeah, I'm not sure. Give it a try.
So
It's
what is
/n/slate. Yeah.
So it's
Slate, I believe.
yeah, SLATE.
Yeah, and then / your username. And
Yeah, no.
make sure
Okay.
you're doing CD, right? Change directory. Change directory, and then you give the flag key. So -p, and then you give the path.
You don't need the -p. This is a no.
If you're in the base, yeah, I guess if you're in the base directory.
Well, I think you need an account with it.
Okay.
I don't have
Yeah.
Slate, so.
Okay. So I mean, so as long as you have space on your base folder in Carbonate, you can be fine. I made a new folder called demo. All right. So sorry, Neil. I'm just gonna okay. So here we can see we finished the download, and it's unsaved as this AGR 00312 _R2.fastq. So we know it's from Illumina, fastq. If we didn't know before, we looked it up. But and then GZ just means that it's compressed, it's a zipped. So the next thing we're gonna want to do is we're gonna use module load. I can type. And we're just gonna fastqc. And so it tells us that it succeeded, like it tells us, hey, in order to do that, I had to load up Perl. So I loaded up that version of Perl, and then I loaded up this. This is the version of fastqc that I loaded up. If it ran across an error, it would tell us here, but it said that it loaded, great. So
the people who are using a local computer are not affiliated with IU.
you can download, you can search, and you can download your own. You can download fastqc to your own local version, and then you run through the README to do the own installation process. Just note that it'll probably take your computer longer than it's going to take the supercomputer to do this. All right, so is everyone here? Okay. No, nope, you have to keep going now. Started. I need this live updates. There we go.
We good?
Okay. All right, so we've loaded the fastqc module with module load fastqc. Yeah, probably. Okay. So for those of you don't know, if you just so if you type in the thing and just give the help, it tends to say most people tend to have like a list of like the things that their module can do. So fastqc is a high throughput sequence quality control analysis tool. So it has one has one command, that is this fastqc, and you just give it like the sequence files. It can take any number of sequence files and analyze and then perform the operation on them. The -O is so you can give it the these are the different options or flags you can give them. So -O is the output directory. So where do you want it to store the results? -F is say fastq, BAM, or SAM. -C is a contaminant file. I don't actually know what those mean, so you'd have to go through their and then additionally, so it gives you a description, and then here are the rest of the other flags you can give it. So anyway, we're just going to run this. Sorry, I don't remember the name of this command. Okay, so we're going to do fastqc, and just going to run it on. It helps if I type it correctly. So fastqc, and then we're just going to give it the name of the file that we want to run. We press return, and we can see that it will start the analysis. Give the output in specific. In this case, it will just give me the output in the same directory. Yeah. So by default, it'll put it in the same directory wherever the file, wherever it was ran in, actually. Especially 10% complete. If you don't want to see that because you have multiple files you're fastqcing, then you can give the flag q quiet -q, and that won't show you this status. And so you can just work on something else while that is happening in the background. And to do that, that is Control Z and then BGs for background. So the job is happening in the background. Okay. Yeah, so we're just gonna let it run for a bit. Yeah. Do you want to pull up a fastqc page perhaps, and we can tell at the what's the file? It's taking any time. This file for next workshop. Yeah. Yeah. Oh, go for it. Yeah. So the R2 means that's replicate 2, right? Yes. What happened to replicate 1? We are ignoring replicate 1. Okay. Because I don't want to go over paired and the differences, and so it's a little more involved. When we're doing the alignment, which we'll get into the next workshop, the different command we have to use. We're just not going to use the. We're just going to use it for the paired int. We'll just use single. We will assume that it's single int. Yes. It's not, but we'll make it. We'll pretend that it is. Yeah, because you can't use a project. Other than trial and error, no. I say it's Cause it's just like any like parameter, just like parameter tuning for like machine learning. Like if it's too little, like you'll end up with too many fragments because it won't, because it won't. Sorry, if it's too little, so you can end up with too few fragments because it'll just try to put everything together. If it's too long, then you end up with too many fragments because it can't put things together. So it's tricky. You just have to mess around with it and then converge to this one. This one seems like the most reasonable. All right, so it's complete. So I press the ls, which just gives you the list of all the files in the directory, and we can see. So we still have our base file. We also have this fastqc.zip, which is just an archive of containing both this HTML link as well as the other different files. So, but the most important one for us is this HTML link, because it's just like a summary of all of the other things that are contained in the archive. So here's where we're going to use our, I'm going to let Neil do this because you have to put in your password and stuff. So, okay. And drag and drop it. You say refresh. Do I just synchronize? Okay. Okay, great. So here I want to just take the okay. So I'm just going to copy this HTML. I'm going to move it over here. And so it'll copy the file from the location where we performed it, and then we just copy it into the directory that I have over here. So specifically for Neil, it's at that directory. All right, you will have it in 2 places. Okay.
Make it simpler on the desktop.
All right, so then we'll just go to the desktop and we'll just double click on here, and it opens up an HTML page with the things that we were looking at. So you can see, so for the summary, it largely will give you like, green checks mean yay, exclamation points mean maybe you should look at this, and red X's mean bad. So so base statistics, it will just tell us so what the name of the file type was, what's the encoding for it, how many sequences were contained. So this tells us there were a total of 228,245,651 different sequences contained in this one file. 0 were flagged as poor quality. The average sequence length was 101 nucleotides, and %GC content is 48%. So we can just kind of scroll down through the various sections. So this is base sequence quality. So during the during the analysis, during the sequencing, sometimes because it's being done multiple times, you can get confidence to how confident are you that the nucleotide that you report at this position is correct. And so here we can, so this is a distribution of the quality scores that are associated with each read, specifically for each nucleotide at each position for each read. And this is just like an average of all of this. So we can see here, it's very confident in basically every nucleotide in every position. And that makes sense because, like we said earlier, Illumina sequencing does the shorter fragments a whole bunch of times. So this makes sense. Per tile sequence quality. So this is similar to what we were just describing this time. It's just going to do a little bit more averaging, and it also swaps the. No, it doesn't. Okay. So again, so it's just doing like the average at each position, and then this time doing some grouping. Okay. Here we have per sequence quality score. So in addition to each position having its own quality score, each read also has a quality score associated with it. So here we can see that most of our reads have a quality score of, let's say, between, let's say, about 36. Right. So this is per base sequence content. So this is the percentage of each nucleotide in each position on average in each read. So this one's giving us a warning because it's you can see that like the amount of it just has a lot of A's relative to the other, like to the others, like very consistently. I don't say it's not saying that that's a problem. It's just saying that's it's not like normal for like human samples, and that might be indicative of some type of problem with the sequencing. In this instance, it's probably not. It's probably just indicative of it not being a human sample. So per sequence GC content, does anyone know why it looks for GC content specifically? So there it says, so there are 4 bases in DNA, so A's and T's and then G's and C's. So why is it talking about G's and C's? No. Well, sorry. Are they more they are more stabilizing for the molecule, for the DNA molecule, but they are in fact long-term less stable, because when G's and C's get methylated and then they get read as A's and T's. So over a prolonged period, so if you have so you just have a single molecule of DNA, and it's all G's and C's. If you let it sit over a prolonged period of time, it will become all A's and T's in a biological organism, not if you do that outside in a vacuum. So the G's and C's, if they're being maintained, that means that there's some selective pressure. There's some reason why the cells are keeping them as G's and C's. So CPG islands are common markers for genes. So having elevated GC content just tells us we have a lot of genes relevant in this in our thing, and that makes sense because we're doing RNA-seq. So we would anticipate that all of our reads would correspond to genes, and thus would probably have more G's and C's than what we would expect. So per base N content. So if it doesn't know, if it can't determine what a nucleotide is, like suppose it gets, in this read I got A in this at this position, at this read I got C at this position, G or T. So it will mark like that it's confused, and N is any nucleotide. Yeah, any nucleotide can go here. I don't know. No idea. Conflicting results. No idea. Just N. So here it's telling us it has no N's across here. They all have some nucleotide associated with them. So this is sequence length. So how long are our reads? Again, from Illumina, we expect them to all be about, all about the same. And here we can see, yeah, they're 101. They're all 101 nucleotides. All right, sequence duplication levels. So here's where it's giving us a flag. And as we look through here, we can see that's because we have a bunch of reads, let's say a bunch of sequences that are repeated. So this would be would be a problem if you were doing like DNA-seq, because we would it would be indicating that like you sequenced like the same section over and over and over again, and that's not helpful. But this isn't a problem for us right now. Why is this not a problem? Like, why can we ignore this warning? So it's saying that it's getting a lot of our sequences are duplicated. There are a lot of our sequences that are exactly the same. True, yeah, this is true. So that's that's an interesting argument, yeah. So like the genome is shorter, and so that's true. So right, so transcripts are going to be less varied than DNA, because like transcripts only serve not serve fewer functions, but are going to they're going to be repeated. Not all transcripts are going to be expressed the same amount. Like you're going to have a couple of transcripts that are expressed a lot more than others, and so we're going to see more repeated regions. Just that there's just less sampling space. Okay, so that's not actually an issue. We don't have any overrepresented sequences. So despite us having more sequences that are duplicated, we don't have any that are over duplicated by a significant margin. We don't have one that's just like overwhelming everything else. Imagine getting 100 million reads and 35 million of them are the same sequence. Well, that's not actually helpful. All right, so then the last thing it checks is this adapter content. And that tells us just where the adapters are in our sequence, as well as tells us what type of adapter they're found. So here we can see that we have this Illumina universal adapter that we see it starting at like 90, and then it goes to about 60. So it's about 30 nucleotide adapter. And so we will have to remove that before we perform alignment step, because otherwise we're not going to get any result, or we will get some results, they'll just be less good. So that's what we'll go over the next section. What questions do we have before we wrap up? So an adapter is a small portion of DNA, typically about 30 bases, that is attached onto the DNA segment as part of the sequencing process. So in order for Illumina's process to work, they have to apply adapters onto the two ends of the DNA. Yep. Because one side of the adapter gets cleaved during, yeah. Yep. Yep. Good question, though. Yeah. Yeah. Okay. So sorry, we're talking about this. So what this is saying is that we have okay. Sure. So so in DNA sequencing, the chromosome, I say the chromosome, each chromosome's 100 million, 100 million characters. So if we were just randomly sampling the chromosome, we would expect to get largely unique sequences. But if for this particular data, we're not analyzing chromosomes, we're just analyzing transcripts. And so there are significantly fewer variation in transcripts than in chromosomes. Does that make sense? So because we're just looking, we're getting a whole bunch of reads from a smaller section of space, we anticipate there will be more redundancy and more duplication than if we try to analyze chromosomes. Does that make sense? Okay, well, that's all I have. Thanks everyone for showing up and bearing with us through these technical difficulties. We have the next section next month where we'll be going over, we'll actually trim the adapters, and we'll go over some of the other, some more hands-on experience with some of the other tools. Okay. Thanks everyone.